Okay, does that sound all right?
Yes.
Perfect. So some of the other talks, including the one we just heard, and the talk from Margaret yesterday, is a great lead into the kinds of things that I'll talk about now. My lab is particularly interested in how the mechanical and chemical environment surrounding cells affects their behavior. And today I'm going to talk specifically about the work we've been doing looking at how cells form networks and form tissue-like structures based on the mechanical environment that they're residing in. So, as I just said, we're particularly interested in how cells interact with their extracellular matrix. There are about 10 to 13 cells in the body. I just recently found out about 60% of those are red blood cells, just a sort of fun fact. And there's about 200 different cells, and each of those interacts with their extracellular matrix in a very specific way to form tissues. And those tissues, of course, come together to form organs. And my lab is very specifically interested in how cells interact with their extracellular matrix to assemble into tissues, and what kind of cues we can give cells to allow them to self-assemble. A lot of the things that I'll talk about are broadly applicable to a number of different kinds of cell systems, but my lab in particular has been interested in the vascular system. The vascular system in the sort of crude cartoon ranges everything from large arteries down to the small capillary networks. In my own work, we focused at all of these scales, actually, but today I'll just focus on the work that we've been doing looking at this process of capillary formation and very small vasculature on the order of microns in comparison to millimeters at the larger scales. So again, we're focused on the smaller scale of angiogenesis, the formation of capillaries, which is, by classical definition, angiogenesis is a formation of capillaries from pre-existing ones. It's this very complicated multi-step process. In this cartoon, it's initiated by the production of some angiogenic factor that's released into the surrounding extracellular matrix milieu that then binds endothelial cells, activates them, causes them to proliferate and migrate out of their pre-existing blood vessel into the extracellular matrix, into the surrounding tissue. The endothelial cells then begin to remodel their matrix. They ultimately form tubes and loops, which then allow blood to flow, of course. And then these vessels are ultimately stabilized by the recruitment of parasites, which is difficult to see, but the parasites basically wrap around the capillaries and stabilize them to support blood flow. And so, again, as you can see, it's this multi-step process, and this is even simplified compared to what we do know about angiogenesis. And my lab is particularly interested in how cells migrate and remodel their extracellular matrix to form these structures, and whether or not we can induce this in an in vitro environment. So we've been thinking about this problem for quite some time. From the time I was a graduate student at Penn working with Dan Hammer up until now when I'm working in my own lab. And we've tried to take sort of an engineering approach to it and looking at each scale that allows cells to contact and form structures. So we started by first looking at how my cartoon cell here adheres to a matrix and the kinds of forces it exerts during that adhesion. Then we moved up and looked at how cells actually spread and assemble cytoskeleton and exert forces during that process to understand the mechanisms by which cells sense their environment, even as they're assembling cytoskeleton and focal adhesions. We then started to look at how the extracellular matrix environment then affects how the extracellular matrix environment affects the ability of cells to migrate, in hopes, again, of a precursor to tissue assembly, which is now the stage we're at. So now we're trying to understand what drives cells to actually contact and connect with each other to form stable structures. And so really, in my talk today, I'm going to focus on these last two steps, how the extracellular matrix environment, both the mechanics of the environment and the chemistry in the environment, affect the ability of these cells to migrate and ultimately form contacts and to self-assemble into structures. So this is sort of old hat after the things we've been hearing about over the past 2 days. The idea that cells can respond to both mechanical forces and chemical forces to change their phenotype. And so there's typically believed that there's some mechanism by which cells sense these cues. They translate them into some chemical signal, some signal transduction pathway that then interpret these cues into a change in cell phenotype. And the right-hand side here, these chemical cues are pretty well understood, pretty well studied. And then the mechanical forces, again, as we've been seeing over the past couple of days, are really coming to light more often, just recently. when people study the vasculature and they think about mechanical forces, most people jump to fluid shear stress. And I won't actually talk about fluid shear stress today. That's not to say it's not important in the system that we're talking about. It's just not the mechanical force that we're most interested in. Instead, I'm going to focus primarily on the mechanics of the matrix, which seems to run in theme with a lot of the things that we've been thinking about. So my lab loves time-lapse microscopy, so I'm going to show you a lot of movies today, which hopefully should convey a lot of the message that I'm trying to get across. So these are endothelial cells, bovine aortic endothelial cells plated on just tissue culture styrene, polystyrene, and these are the kinds of things that it's better than TV. So we watch the cells migrate around, and this, again, you would see this in any common culture dish. This is what's happening when your cells are in the incubator. They move around relatively randomly. These cells divide once a day or so. They move by the ruffling of their leading edge. And again, under no stimulus, the migration is somewhat random. And the important part here is that in culture, of course, they're not assembling into any sort of structure, except that they're attached to a dish, maintaining a well-spread morphology. And in fact, any amount of quantitative analysis on these dishes has led us to believe that as far as cell-cell contacts are concerned, they're not really affecting the cell behavior in a marked way.
Do they cluster if you wait on?
Do they, say that again?
Do they form clusters?
No, not these cells. So that's the one caveat I'll say. So it's very different than epithelial cells. These cells do not form, at least not in our hands, not ever form clusters. When they divide, they might bump into each other, but if you analyze enough cells, we don't see any sort of directed cell-cell adhesion. The migration is relatively random on very stiff surfaces, and that's where the story is going, how it changes when you change the mechanics of the environment. So what we were actually intrigued by is a number of studies, largely started by Judah Folkman, looking at how angiogenesis occurs and creating in vitro systems to look at this. And essentially, if you can take endothelial cells in culture and place them on the correct extracellular matrix environment, they'll start to self-assemble without any other external cues. So even independently of growth factors or cytokines or anything like this, the cells will start to self-assemble and form these network-like structures that branch out and even form lumens. And so what we were interested in understanding is what about this environment is inducing this behavior. So it was largely thought that it's the actual composition of the matrix that you're putting your cells on, whether it's a matrigel laminin or whether it's a collagen or whether it's a fibronectin is going to induce these changes in endothelial cell assembly. And we actually speculated, because of these gels being relatively soft, that in part that it's not just the chemistry of the gel, but it's also the mechanics of the gel. And so I'm going to talk about that, what led us to that thought and where we've gone with it since then. So a lot of our work is built off of this differential adhesion hypothesis, which some of you might be familiar with. It was first postulated by Malcolm Steinberg in the 60s. And basically, he developed a hypothesis to describe how tissues form in early embryonic development. And the idea is basically that tissues form based on the strength of cell-cell adhesion. So when cells are more tightly bound to each other, they're more likely to stay bound to each other, and more weakly adherent cells will then surround those tissues. So it's basically minimizing the surface free energy to maximize the cohesiveness of the structure. And he developed the theory basically by taking embryonic tissue and digesting it into the multicellular types and then allowing it to self-aggregate. And what he found is that cells that are most strongly adherent to each other tend to form this core of the reaggregation. And cells that are less strongly adherent to each other tend to form around these structures. So again, the idea is based right in the title that this differential adhesion that cells have for each other allows cells to form structures. And so we started thinking about this and thinking, well, if it's not only cell-cell contacts that can cause this behavior, but maybe it's also cell matrix contacts. So how strongly adherent a cell is to its matrix versus how strongly adherent a cell is to another cell that will prompt its self-assembly and sorting. And so we started off in a very simplistic way. Hopefully you guys can see this. It looks a little dark up there. But we start off in a pretty simplistic way of basically watching cells migrate and trying to figure out what goes on when they come into contact with each other. And we saw three fundamentally different behaviors when we watched cells come into contact. The first is this first behavior. You can see the two cells right here. And basically, there's nothing really significant. This is the same sort of behavior you see on polystyrene, nothing too special going on. The cells migrate. They do touch from time to time, but there's no significant shape change, so the overall projected area doesn't change. There's no significant cell-cell contact attachments that keep the cells bound together. The second behavior we saw was actually very, very different. And we've termed it sort of this tug-of-war behavior. You'll see these two cells. What happens is the cells come into contact, and then they form basically a tether as they pull apart. And then one basically loses the tug-of-war and will actually get popped off the surface and sort of flung in the opposite direction. So if you watch, so they form this tether, and then they sort of fly apart. And then the third behavior is somewhat like this, except what happens in this behavior is that the cells are less adherent to their matrix. They come into contact, they again form this tether, and again the tug-of-war ensues, and one cell gets pulled off the surface towards the other and loses the tug-of-war. So you can see it a couple times. The cells will try and respread on the surface, they'll form this tether, and then one cell will get pulled off the surface. And so there's this constant tug-of-war between the cells. And these three behaviors we see pretty much exclusively. So when you watch cells come into contact, depending on the environment that you're giving the cells, one of these three behaviors occurs. And so we looked at these behaviors in the context of the kind and amount of ligand that we're giving the cells. So independent of mechanics yet, we're simply looking at how the density of the extracellular matrix that the cells are given, how that affects the cell contact behavior. For these experiments, we're using a simple peptide, an RGD peptide, which binds integrins, but doesn't have all the complexities associated with fibronectin or collagen. It's just a simple integrin binding sequence. And what we found is, just to go over, this A behavior is where cells come into contact, but there's no real significant changes in the cell behavior. This B contact, where the cells come together, they form a tether, they have this sort of tug-of-war issue, and then one cell ends up getting snapped off the surface away from the adjacent cell. And then the C-type behavior, where the cells come into contact, they form a tether, and then one cell gets pulled towards the other cell, losing the tug-of-war. And so what we found actually is that this behavior is highly dependent on the amount of protein, the amount of adhesivity that you give the cell. So the more adhesive a surface is, the more likely it is to exert this A-type behavior, where the cells stay well spread and the cell-cell contacts have no real effect on the cell behavior. And the less adhesive the surface is, the more likely the cells are to come into contact and get pulled off the surface in this tug-of-war, where they basically are trying to make a decision between cell-cell contact and cell-substrate contact. And in this case, where there's very little ligand, they almost always make this decision to be connected to an adjacent cell rather than to the matrix. So we sprung from that and said, okay, so if we're making a surface less adhesive using chemistry, can we also do it using mechanics? So it's sort of well established now that if a surface is more compliant, cells tend to spread less. Just like if you give them less ligand, they tend to spread less. And so we looked at the behavior of these cells on very compliant substrates to investigate how cell-cell contact varies based on the compliance of the substrate that you're giving the cells. And what we found is that on soft substrates, the cells tend to stay stuck together throughout the duration of the experiment. So this is a 6-hour time course, but if you would watch these cells for 12 hours or 24 hours or 48 hours, assuming that they don't divide and assuming no other cells come in the field of view, these cells stay stuck together. So
do they migrate together, or they just, at rest?
I'll show you data about that in a second, but they do move some. So you can, if you even look here, you can see that this cell has changed shape substantially from 0 hours to 6 hours. So they're moving somewhat, but if you look at the overall translocation, it's relatively minimal, whether they're alone or whether they're with a cell pair. Let's skip down the stiff substrates. So there's no surprises here. If you give a cell a substrate that's really stiff, identical chemistry as the soft substrate, but just stiffer, the cells will bump into each other and then migrate on their merry way. So it's almost as if the other cell wasn't even there. And the really interesting behavior actually was on these intermediate compliant substrates. What we found actually is that the cells will come into contact, and then they'll separate, and then they'll come into contact again. And this little time course I've shown you is 6 hours, but in fact, over the 6 hour time period, these cells have come into contact and separated over 6 times. So about once an hour, they'll touch each other and then move away from each other, and then touch each other and move away from each other. And we've seen this behavior pretty consistently, and this will continue to happen, in our experience at least. This will continue to happen until, again, either the cell divides or another cell migrates into the field of view. So with no other external influences, these cells come together and separate, come together and separate, and come together and separate. So this was a pretty interesting phenomena, in our opinion, because it's not the same as clustering, right? So it's not as if the cells are just proliferating and moving outward, but in fact that they're migrating away from each other and then in a very directed fashion migrating back. And so as promised, we looked at the migration of these cells. Here we've quantified the dispersion, which is basically a measure of the cell diffusivity, how likely a cell is to move away from its starting point over time. And let's focus first on the dark bars for single cells. As we might expect, cells are able to migrate better on stiffer surfaces. And this has been shown by a number of groups. Varies a little bit by cell type, but in general, cells are more migratory on stiffer surfaces. Now, if you look at the cell pairs, let's start with the stiff substrates. On really stiff substrates, the behavior of cells interacting as pairs, the migratory behavior of cells interacting as pairs, is not statistically significant from the behavior of a single cell. So again, it's as if that second cell isn't there. Let's go down to the really soft surfaces. Again, the behavior isn't very significant, is not significant, but then again, our cells aren't moving much at all on soft substrates to begin with. And then again, this is basically our way of quantifying the images I just showed you, which is that single cells migrate well, fairly well on these intermediate compliant substrates, but when they interact with cell pairs, they don't migrate very far from their starting point. So again, it's our way of quantifying the fact that when cells are interacting as pairs, they tend to stay in the same location, the same vicinity as their partner cell. So we continued to pursue this because it seemed pretty intriguing to us what was keeping these cells together. And we actually quantified what we considered our region of influence. So how far away from the cell edge can a cell actually propagate forces and displacements that the second cell might actually do both sense. So the overriding question here is a cell exerting traction forces on its substrate that are actually inducing adjacent cells to come closer. And so we started off with a really sort of back-of-the-envelope way of doing this. We looked at a cell, and we looked at the displacements of beneath the cell. The substrate is basically embedded with beads. I'll talk more about this in a minute. But the substrate is embedded with beads, and then we just tracked the bead displacements, and we looked to see how far from the cell edge these beads appreciably move. So of course this is limited by our imaging resolution, but it gave us sort of again a back-of-the-envelope guess on how far the disturbances the cells are creating move away from the cell edge. And what we find, so let's start with these gray bars, what we find is that on soft substrates, the displacements are relatively far from the edge and then decrease as you increase the stiffness of the gel, and that makes sense. On really stiff surfaces, it's not even possible to see the substrate displacements because they're so appreciably small, if existent at all. We then looked at how far the cells actually separate from each other. So remember I said the cells touch and they come together, they touch and they come together, and we just measured the distance that they separate during this phenomena. So how far can they possibly get from each other? And what we found actually is that the cell-cell separation distance over our time period, which is again 6 hours, is very large on stiff surfaces. And on soft surfaces it's very small, so the cells aren't moving at all from each other. And then on these intermediate compliant substrates, the actual distance that the substrate displacements is very comparable, statistically similar, to the distance which the cells separate. So this data indicates to us at least that the cells are pulling on the substrate and the cells never move further from each other than the distance of this pulling mechanism. So it led us to this question now, whether or not cells can actually mechanically communicate through the substrate. So can one cell pull on the carpet and have another cell sort of get slid towards it? Can it sense the tension created from one cell to the next? And this actually wasn't a huge stretch. A lot of the data collected by Yuli Wang in his group has indicated that cells can sense tension in the substrate. If one uses a microneedle and pulls on a substrate, cells tend to move towards the increasing tension in the substrate. However, it's never been shown on the scale at which cells exert forces. So can cells actually use this as a mechanism to come together? And so to figure this out, whether or not cells can actually sense each other, we used traction force microscopy, a version of which Margaret talked about yesterday. We used the elastic substratum method as well in collaboration with Micah Dembo. And the general gist of it is that we take our deformable substrates that are made out of polyacrylamide, we embed it with fluorescent markers, we watch the fluorescent markers move, and based on those movements, we back out the forces the cell exerted to create the substrate displacements. And again, my love of time-lapse microscopy. So this is an endothelial cell migrating on a polyacrylamide gel embedded with beads. So you can see the beads somewhat in the background. This is the corresponding fluorescent image. And again, you can see the beads underneath the cell moving. So a little bit like footprints in the sand, so to speak. And we can track those bead displacements and then back out the forces the cell exerted to create those bead displacements. The reason we wanted to implement traction force microscopy in this case was to then understand whether or not cells could exert enough force that an adjacent cell might be able to respond to those forces. So this is just to get us all on the same page. We've heard a lot about contractility of cells. Endothelial cells, too, are very contractile. This is an output of traction force microscopy. Basically, cells tend to exert the largest forces at their cell edge, and all those forces tend to point inward. So the cell is very contractile. It's a little bit clearer here on the color contour plot where these warm colors indicate the magnitude of the traction that, again, is highest at the cell edge. And not just at the cell edge, but on the extensions of the cell. And so we wanted to use traction force microscopy to look at how cells change their traction forces as they're coming into contact and determine whether or not a cell can exert enough force that it can actually propagate through the substrate, the displacements can propagate through the substrate and then be felt by an adjacent cell. And so what we did was we looked at many, many cell pairs as they were coming into contact. This example I'm showing because if you spend a lot of time looking at cell migration, you would guess that this cell is probably moving from left to right, and this cell is probably moving the opposite direction from right to left. And so we would expect perhaps that these cells might not actually come into contact at all if they were moving at about the same speed. And what in fact you see over time, and this takes about 2 hours or so, what happens is that the cell will actually start migrating towards an adjacent cell. And so if you look here in the first frame, you can see the cells sent out a very small pseudopodia towards the adjacent cell. What's interesting is by the second time point, this cell has started to extend some of its membrane towards the adjacent cell as if it's going to now reach out and touch this cell. And by our final point, we do have cell-cell contact where both cells have reached out their arms to come into contact and touch. And so again, we chose these because the movement that we end up seeing is sort of counter to the movement that we would have expected to see. And so the question is the presence of an adjacent cell prompting these cells to come into contact? And so we use traction force microscopy to look at the forces that cells are exerting during this change. So let's talk about the most obvious thing. First of all, the cells, before they come into contact, the most significant forces are all localized to the cell edge, just as we've always seen. And as the cells come into contact, actually, there's a really big drop-off in the force in the middle of the cell. So basically, once they've come into contact and formed a stable cell-cell contact, it looks to us, at least, like the contractile balance of the cell shifts all to the cell periphery as the two cells now balance each other. But what we're actually really interested in, again, is whether or not one cell is exerting enough force for an adjacent cell to feel that force. And so we focused particularly on this pseudopod because this is the first guy to reach out and touch someone, so to speak. And this guy responds to those forces. So we're going to look at the force that this pseudopod exerts to determine whether or not those forces are substantial enough for this adjacent cell to feel those forces. And what we found actually is just using some basic elastic theory that the force of the pseudopod, again, well, let me cut to the punchline, that the force that this pseudopod exerts is significant enough to be sensed by the adjacent cell. And the way we approximated this was, again, to calculate the force the pseudopod was exerting using traction force microscopy. And we looked at basically the distance between the two cells. And then we modeled this cell and its ability to sense an adjacent cell based on whether or not 2 points or 2 antennae on the receiving cell would shift relative to each other, right? So if there was some relative motion within the cell, that it could then sense changes in its substrate. And so we approximated that as the length of a covalent bond, so basically an integrant attachment. And the separation distance between these 2 antenna points is about 10 nanometers, which we're approximating integrant clustering in a focal adhesion. And so what we determined using all of this information, in addition to the elastic modulus of this particular gel, is that the distance of significant transmission for this case and others is about 30 microns, and the distance between the cells at that time point is about 25 microns. So it indicates to us, at least on a back-of-the-envelope calculation, that this pseudopod is exerting enough force to create displacements under an adjacent cell that could actually be sensed and responded to by an adjacent cell. And so, so at least for this part of my talk, we believe that cell-generated traction forces can actually drive cells to come together. And so, again, this is just us marching up, trying to understand now how to use mechanics to control tissue self-assembly. And so we started first by just looking at these same regions of gels that we've been working with and looking at how now cells interact when there are multiple cells on a given substrate. And the interesting thing about these pictures, which I'll describe in a second, is that the only thing that's really changing is that there's an increase in substrate stiffness. And so the substrate is getting stiffer and stiffer, but it's the same collagen that's put down on these substrates. So we're not giving it any external growth factors or chemical cues. We're not mixing in matrigel. This isn't 3D, it's just on 2D surfaces. And yet what we find is that as you make surfaces more compliant, the cells are more likely to start self-assembling into network-like structures. As the substrate stiffens, they start to look like they do on polystyrene. So they tend to spread out, and there's no obvious directionality to how the cells attach to each other. At intermediate confiances, we see an increased cell-cell connectivity. So you can see that they're touching more than you might expect on polystyrene, but not to the degree that we see at the really soft surfaces. And so now we're going to probe this mechanism a little bit and see how it works. And so the first thought was, well, maybe the surface adhesivity is decreasing, and that's actually driving cells to connect. And so now we did the same experiment, and we changed the surface, the matrix mechanics, but now we also changed the amount of ligand down on the surface. So in these experiments, we took the amount of collagen from the previous experiment and went down 1 order of magnitude. And what we found is that on really soft surfaces, if you decrease the collagen content too much, the cells can't stick anymore. So the combination of soft plus low ligand density equals cells don't stick. But as you move up, what we see is that the cells regain some of this connectivity where we wouldn't have seen it otherwise. So this is a 10,000 Pascal gel where if we had a lot of collagen down, they would be behaving like we see on polystyrene. But on fairly, I'm sorry, that's labeled here, this is 10,000, not 5,000. On a 10,000 Pascal gel where we normally see no substrate adhesivity, or sorry, no network formation, we suddenly start to see cells preferring cell-cell connections over cell-substrate connections. So if you modulate the chemistry versus the mechanics, you can start to induce some amount of cell assembly. And so so what we actually did was to try and understand how these networks were forming. We did a lot of time-lapse microscopy to look at this actual behavior. So what's inducing these cells to actually self-assemble? And so what you can look at here, and it's a little bit hard to see, but if you take any cell that's sort of reaching out and you start to watch it, what happens is the cell starts reaching out, and eventually you'll see on this side, these cells will start to reach out too. And then eventually they'll connect up. And this is going to happen, I think, over here next. The cell's going to reach out, and then somebody over here is going to get a signal to do the same thing. And then they reach out over long distances, and they connect up. The one thing, too, you'll note in this video is that proliferation doesn't stop. So eventually, some balance gets struck where cells will just start to fill in. So this is a very transient structure. It's not permanent. It doesn't last forever, but cells will start to assemble and eventually close in on the surface. But in the meantime, they form these odd structures where they're connecting over long distances. And we're working very hard to understand the mechanisms that mediate that.
So initial situation was homogeneous.
Say this again?
Initially, you placed cells in homogeneous.
Yes. Yes. So cells are actually surprisingly sensitive to light before they're well adherent. So it's very hard to capture this from time equals 0 from the time they hit the plate. But we wait. This is about 24 hours, I think, after plating, and then we wash them from that point.
What is the threshold? What is the maximum distance that you never see that the cell will be able to make a connection? Or they already make a connection even if they are far apart? Yeah.
So that would be a hard measurement to make, but we can make the opposite measurement. So it's hard to say what the maximum is because that's a difficult experiment to run. But I can tell you what we've seen. It's hard to make the measurement of the threshold. Experiments. I haven't really thought about that. It would require probably some fabrication or patterning of the protein. But what I can say is that we've seen cells come into contact over hundreds of microns.
So what about chemical messaging between the cells? If they're more sensitive to contact and you don't have as much RGB on the plate, then that would maybe suggest that there's something not mechanical that's going on between them?
That's a good question. So I'm going to talk next about the other factors that we've seen that help stabilize these networks. But as far as like a chemotaxis cue, for instance, where one cell is releasing factors that the other cell is responding to, we've found it hard to control for that because even really low, say, fluid shear stresses to wash media away affect the ability of cells to attach, and they affect the migration of the cell even independently of chemical factors. What we have done is we've switched out media. So we've taken media from cells on soft substrates and put it on stiff substrates and vice versa to see if there's something being released in general due to substrate compliance that's affecting the ability of cells to come together. And we haven't seen any differences in the cell behavior in those cases.
Sorry, what about really thin membrane tethers between the cells?
Oh, that's a good question, too. So there was this, perhaps you've seen a paper, there's a paper, I'm getting older, so it's probably, I was strolling with the math like 6 or 7 years ago now, with these, I think they're called the TNTs, nanotubes that connect cells, and they're actually very hard to see because you can, and to be honest with you, we can't really control for that. So the highest resolution microscopy we've done, we haven't seen anything that connects the cell, and then nothing with immunostaining, but presumably if they're that thin, any process like that is going to wash it away anyway. Okay, so we're trying to understand what actually mediates this process of this network formation, whether or not we can actually predict it based on how cells are adherent to the matrix. This is well known for a number of cell types, and this is the threshold for endothelial cells, the fact that as you increase the stiffness of the matrix, you increase how well spread the cells are. So on really stiff substrates, the cells are pretty much plateaued out at their maximum area, and on more compliant substrates, the cells are less spread. And so what we actually found was that if you take the cells subconfluently, before they formed networks, or before they formed confluent monolayers, and you look at the ratio of area to perimeter. So if you think about this, it's basically a measure of how round or how convoluted the cells are. You can then predict network assembly based on that. So let's walk through this a little bit. On the empty bars, the white bars are 100 micrograms per mil of collagen, which is a relatively high density, and the black bars are 2 orders of magnitude less than that, 1 microgram per mil. So again, this is the surface where we're seeing networks on very soft substrates, and this is the density where we're seeing networks on very stiff substrates. And so we looked at this now against Young's modulus. We can't measure the cell area on soft gels at this low density of collagen because the cells aren't sticking very well. But what we find actually is that this ratio of area to perimeter is a predictor of whether or not the cells will form networks. Again, this is before cells have formed networks. This is just subconfluently. And what we find is that if you look at the ratio of area to perimeter, when it's above a certain threshold, the cells will not form networks. And when it's below a certain threshold, they will form networks. And I have to say this line was drawn without any sort of statistical test. It was more qualitative. When we see a higher area to perimeter ratio, indicating that the cells are more well spread and a more rounded morphology, they don't form networks. And when they form a more spindle morphology, where the perimeter is higher relative to the area, they're more likely to form these networks.
You're talking about the area and perimeter of the network, of the cell collection, not just of one cell.
Of a cell.
Area to perimeter of a cell.
Mm-hmm.
Okay.
So we started looking at how the traction forces change on these substrates where the cells become very polarized and spindly. And what we find is that the tractions are very much localized to these cell edges. So it says to us that the cells are able to target their traction forces and their contractility in a polarized way, pulling on the substrate basically from only two directions rather than from all various directions. And this is some really interesting data that we're still sort of thinking about. So the convention is that force increases as the substrates get stiffer. And we've done these tests for endothelial cells now, and we found pretty reproducibly that as you increase the substrate stiffness, the force actually decreases. So the force is decreasing where cell networks tend to assemble, but the contractile force, and this is of single cells, is really high at the stiffness where we see networks assembling. And so this data we're still sort of toying around with a bit because it actually contradicts what people have seen for other cell types. So we think that it's probably very specific to endothelial cells and this mechanism by which cells are driven to connect based on the stiffness of the substrate that the cells are sitting on.
What's the strain in your matrix that you're measuring? And the forces you measure are in kilopascal, or several hundred kilopascal.
So the forces for these soft substrates are very, very high. So the strains are even hundreds of microns away from the cell edge.
And are you worried about your force reconstruction in that?
So yes and no. So we've gone back through and been very careful about whether or not, when we track our bead displacements, whether computationally they match up with what we see experimentally. And so far, everything seems to match. We can't go softer than this, though. So the networks always form at 500 and 200 Pascal gels, but the displacements of those are impossible to track. So as I promised, I would talk a little bit about how the chemistry of the environment affects the cells. And there was recent data actually in 3 dimensions that fibronectin is required to stabilize angiogenic structures, and again, in 3-dimensional matrices in vitro. And so what we did was went back and said whether or not fibronectin was actually required for these 2D structures that we were now forming. And what we found was that, it's a little bit hard to see, but this is, the left panel is where networks are forming, and the right panel is where the cells are confluent, so soft substrates versus stiff substrates. And we stained here, this is Floyd and Daffy, just so you can see where the network is. And then we stained for fibronectin, and what you can see is that when the networks form, the fibronectin colocalizes right on top of these networks. So we obviously don't see it out where there are no cells, but more than that, it tends to form fibrils to directly align with where the cells are. And this is in contrast to where you see these monolayers. It forms more of like a web-like structure rather than these parallel fibrils on top of the cells. And so since we saw fibronectin aligning with our structures, and this is fibronectin not that we added, so our substrate is not coated with fibronectin, it's coated with collagen, and we're not adding any exogenous fibronectin other than what's in the serum. We then asked the question whether or not fibronectin was actually required to stabilize these structures, right? So where's the balance between driving the cells together using mechanics and keeping them stabilized using fibronectin? And what we found is that the cells don't actually require exogenous fibronectin to assemble in a network. Now, by exogenous, I mean the fibronectin in the serum. So in this experiment, we took out the fibronectin from serum. We redid the experiment, and we found, again, fibronectin still colocalizes with the cells. So because we're not adding fibronectin, we're taking all the fibronectin out of the cell, the conclusion is that this fibronectin must be fibronectin that's actually produced by the cell themselves. So the cells are producing fibronectin and using it to stabilize these networks. But this still doesn't answer the question whether or not it's actually required to do the next experiment. We added an inhibitor that prevents fibronectin from being polymerized. So the fibronectin can still be produced by the cells. It can still adhere to the cells, but it can't be polymerized into these fibrils that align with these network structures. And we find something really interesting, which is that this is a time course over 17 hours. And what we see is that this is a compliance where we normally see network structures form. Over time, they tend to start aligning, like we would expect. And then over additional time, they just disassemble. And the cells aren't dying, but they're not adhering, they're not forming networks. And we stain for fibronectin, as we'd expect. It's not forming these fibrillar structures. It's adhering to the cells, it's being produced, but it's not actually being polymerized. And so our conclusion, at least from these experiments, is that the mechanics can help drive the cells together, as we can see in this experiment. But then the maintenance of these stable cell-cell contacts into these network structures requires fibronectin to stabilize the structures and to keep the cells in contact. And so, in conclusion, I hope to convince you that we can use compliance and mechanics to promote cell connectivity, and we can use it as a modulator of tissue formation. The way cells actually adhere to this matrix, which we've measured as a ratio of area to perimeter, can actually predict the likelihood of 2D network formation. And so we think that's a measure of substrate adhesivity and how well cells can adhere, and then whether cells make the choice to adhere to each other versus adhere to the matrix. And the chemistry is also important. So in our case, at least, the fibronectin polymerization is required to stabilize these network formations. So cells can communicate through their substrate. They can use these contractile forces to assemble into networks, and then they can use these and then they can stabilize these network structures using chemistry. And so really network formation is this balance between chemistry and mechanics that allow cells to form tissue-like structures. So with that, I'll acknowledge my lab. The latter part of this work was done by Joe Califano, a third-year graduate student in my lab. With that, I'll take any questions.
I have two questions. On the RGD surfaces, do you see any fibronectin being secreted and remodeled on the surface?
That's a good question. So on the surface alone, no. So we've done tests where we've put down RGD, so it's an absence of cells, put down RGD, and then incubated with fibronectin or serum, and then stained. And we haven't seen anything in here. But again, that's in the absence of cells. So we haven't done those experiments with cells.
And my other question is, your work and other people's work makes the coherence be like slaves to the, whatever the integrins or some ECM adhesions want to do. Like, if they can adhere, then all of a sudden the coherence don't do anything useful. I mean, do you have any comment, how would they regulate their own cell-cell interactions without having to modulate cell ECM adhesions?
That's a good question. So I didn't mean to ignore coherence. I love coherence as much as I love
I don't know
integrins.
what they are in these cells. What are the cell-cell
So VE cadherin is the big one. And we've looked at VE cadherin expression based on substrate compliance. So just in subconfluent cells, is there something about the integrins that change coherent expression?
Yeah.
And we don't see any changes. If we then start to look at cells as they assemble, we can see VE cadherin expression start to go up as they form cell-cell contacts.
But even mechanically, they just seem weaker. Like they're not able to maintain a contact in the presence of a strong ECM adhesion.
I think that is what we're seeing. I think that they're weaker bonds than integrin bonds, and I won't be able to cite numbers, but I think Cheng Zhu, for instance, has done a lot of experiments on that, looking at cadherin strength and integrin strength, and that it's accepted that cadherins are weaker bonds. But given the correct compliance, I think coherence play a really critical role in transferring those forces. So when we look at traction maps of cells coming together, the forces look like they get transferred from cell-cell or cell matrix connections to cell-cell connections, and those cell-cell connections can stabilize forces over the body of the cell. So I think they're important, and we're working at getting at what kind of forces they exert and how important they are in this stabilization.
Suppose you put a suspension of cells with low density, yes? So cells are far away from each other on a soft substrate. So you said that after some time a network will be formed. Also, you said that cells almost do not migrate on those soft substrates. So how cells become close to each other?
So there is a caveat to that. There is a threshold level of plating density that you have to have for networks to form. So if you don't plate enough cells, nothing happens. The cells stay adherent. You don't see networks. Eventually, assuming there's enough adhesive ligand down and the cells can proliferate, then the cells will start to proliferate so they get to that threshold density, and then they'll start to form networks. On the other hand, what we typically do is we found the amount of cells we need to plate to get networks to form, so we don't have to wait for them to proliferate up to that point.
One more question.
You said that the stiffness is you looked at between 1,000 and 10,000 pascals. How does that compare with the stiffness of, for example, the extracellular matrix in our bodies or little animals?
That's a good question. So that's obviously dependent on the type of extracellular matrix we're talking about. The 1,000 Pascal regime is the region of interest for tissue formation in general. 1,000 in sub 1,000. 10,000 for endothelial cells. In this context is too high to be physiological. Now, if you now go up to large arteries and you're talking about the extracellular matrix under the large arteries, that starts to approach the, I think, 5,000 to 8,000 range. But as far as microvascular tissue compliance, 200 to 1,000 is about the physiological range we're interested in.
Any more questions? You talked about how one cell can sort of sense the other by pulling on them. And you talked about sort of a strain, like a displacement. But do you have any comments on if that other cell is sensing that strain or like a stress?
Oh, that's a good question. I don't know. We thought a lot about it, and Rumi might be able to answer this better than I can. And to be honest with you, I'm not sure. We assumed in our calculation a strain because we were thinking about integrins again, and we're thinking about relative translocations and movements in the actin. But just as easily could be stress. But to be honest with you, I'm not quite sure how to get at that answer.
I see there's still quite a few questions. Perhaps in order to stay on schedule, you should preserve them for the discussion later, if that's okay. Before we go to the coffee break, Christina Marchetti has some announcements.
Thanks again, first of all. Colleague, we have an important thing to do. I promised prizes for sound bites and posters. So first of all, these prizes are actually cash prizes, and are possible thanks to the generosity of CASE, which is the Center for Advanced Systems and Engineering at Syracuse University. And we have today here David DiMaggio, who handles their co-op and internship program, and will help me here are the prizes. I realize it's possible some of the people getting the prizes might have left, but I hope at least some of them are still here. So David might tell you a few words about CASE, first of all.
Good morning. Can you hear me okay? Thanks for coming to Syracuse. I guess you're here from around the world, right? All I know is the weather got better since you got here. Because it was terrible Sunday, and I know Monday, Tuesday, Wednesday it's been beautiful. It's actually been today also. So thanks for coming to Syracuse, and actually this event is one of the first major events in this building, which has only been open for about 9 months. And my office is right out here, so you go out and get your coffee and you stare into that aquarium-looking fish bowl that says CASE. That's where I can't hide. I run a co-op program, and basically my responsibility is to connect employers across New York State with students at Syracuse University. We've had about 250 students working since we started the program just 3 years ago, and we offer a very unique service in that many of our grad students here, if you're not aware, are foreign nationals, and we have facilitated the ability to pay those students through the university so they can all get out in the workplace without necessarily having to do their practical training and work much earlier in their college graduate degrees. So welcome and thank you. CASE is the Center for Advanced Systems and Engineering, which is sponsored by a grant from New York State, which mainly around data fusion and predictive analysis, areas which I am not familiar with. I'm more of a commercial person that tries to connect people with employers. So thank you for coming again, and we did provide some support here today. So I got drafted to give out a couple of awards. I think the first one is going to go to the first poster session, which goes to Henry Fu from Brown University. Is Henry still here? Henry's the title of Henry's poster was Theory of Swimming Filaments in Viscoelastic Fluids. Did I say that right? Congratulations.
Thank you.
Don't spend it all in one place. And for the second poster session goes to Volker Schaller from Technical University in Munich. And Volker's poster was self-organized patterns in 2D active fluids. I was going to say second because I saw it was second, but it's 2D. So. Volker, congratulations.
Thank you.
Thank you very much, David, and I'll give out the posters for the sound bites, where actually the judging committee had a bit of a harder time coming up with one winner. There were 3 talks that the 3 judges unanimously found they were truly outstanding. And so we actually decided to give out 1 first prize and 2 honorable mentions, which means the first prize gets a little less than the poster prizes, but I think this is just fair. So the honorable mentions go to Xiaofan Luo from Syracuse University, from working in the biomedical engineering department. And I was going to say I forgot, I'm not as well prepared as David was, I have to say. So the title of his talk was "You Can Get Blood from a Plastic and Use Self-Healing Polymer System." Congratulations. The second honorable mention goes to Valerie Cross from Cornell. I believe Valerie is probably not here. Oh, she is. Great. Okay, come down, Valerie. And her talk was Mechanism of Cell-Cell Communication within 3D collagen matrices during vascular network formation in vitro. And the first prize goes to Kelly Berg, also from Syracuse University, and Case Western, Case Reserve University, and also working in biomedical engineering. And the title of her talk was Liquid Crystalline Elastomers as Soft-Shape Memory Networks. And with that, you're all free to go get some coffee. Thank you very much.